I will try to outline some ideas, some concepts, how to improve the Annex-I criteria to bring it in line with the existing evidence. Next slide, but without putting in question the goal of the European Union to adapt the current regulation of NGT plants. So next slide, please. We already heard that this study, as presented today by Dr. Koller, is likely to be the first study to systematically assess the differences between NGTs and conventional plant breeding. This might take you by surprise. But indeed, as already somehow mentioned by my colleague, EFSID did not have the mandate to systematically assess the differences between conventional breeding and NGTs in regard to intended and unintended effects, even explicitly stated something into this direction. So now the cases cited by Dr. Koller include camelina, lettuce, maize, mustard greens, poplar trees, rice, strawberries, sugarcane, switchgrass, tomatoes, and wheat. And it's also shown that the respective genetic changes often are very minor, but the results significantly differ from those achieved through conventional plant breeding. Overall, there's at least now, finally now, strong evidence that the differences between NGTs and conventional breeding, both in regard to innovation and risks, are not depending on number of mutations. Next slide. So I'll shortly summarize what we've heard already. These differences between NGTs and conventional breeding are mostly caused by evolutionary mechanisms. These molecular mechanisms developed by evolution could be called mechanisms for self-protection of the genome, and these certainly make sense from the perspective of evolution. They stabilize the characteristics of the species and its interactions with the environment, while still allowing adaptation. And these mechanisms are very well known and documented from plants and animals. And there are two general levels that can be identified as the structural factors that may nullify the statistical likelihood of specific gene combinations to occur, and also reactive factors such as gene repair processes. Next slide. So the constraints experienced in conventional plant breeding are very often just minor hurdles for NGTs. They can be overcome in many cases, not in all cases, but in many cases. And it's a fact, it should not be put in doubt, that NGTs can be used to develop plants that are new to the environment and are unlikely to be obtained from conventional plant breeding. But and this can be achieved just by introducing minor genetic alterations. So consequently, a case-by-case environmental risk assessment is necessary, taking into account intended and unintended effects. So when it now, next slide, comes to the goals of the current regulatory initiative, we would agree with those goals as stated in recital 14. In number, to ensure legal certainty, the regulation should set criteria to ascertain if an NGT plant is equivalent to natural occurring or conventional bred plants. A procedure should be laid down to verify it and take decisions whether these criteria are fulfilled. And the criteria should be objective and based on science. And now we see the Commission tried to come up with this criteria, next slide, in its Annex-I, which is supposed to define criteria to equate NGT plants with the conventional bred varieties. And here you see what was already shortly discussed at the beginning, triggered by the question of Österreicher, that up to 20 genetic modifications for plants, including substitutions and insertions, up to 20 nucleotides should be allowed. This is considered to be a threshold that describes plants that can be equated to conventional bred plants and would not require risk assessment. And in the current compromise text, even we see the idea that these 20 genetic changes, that can be 20 nucleotides per change, or larger insertions or deletions, systemic elements, that all these 20 genetic changes with all these further up possibilities can be multiplied if a plant has more sets of chromosomes. Such, for example, in wheat, we have 6 or 8 chromosomes, and in under these circumstances, the number 20 would then be multiplied with the number of sets of chromosomes. And all we can say is this is quite arbitrary and confusing. It's not established in any region of the world. It's not established or derived from any scientific publication. This is just bureaucratic monster meant in the end to allow, we would guess, much more than 90% of the NGT plants, which are currently under development, to be placed on the market without environmental risk assessment. So next slide. So when we do a critical assessment of this proposed Annex 1, it's very clear that the Commission overlooked that the relevant differences between NGTs and conventional plant breeding are mostly independent from the overall number of mutations. I think this was very clear from the previous two presentations. Therefore, thresholds for the number of mutations in regard to risk assessments do not make any sense. This, I think, is a crucial finding. Forget about all these thresholds. They have nothing to do with the necessity to introduce a risk assessment. Also, the current compromise text from the trilogue suffers from the same basic misconception of the Commission's proposal, and I will shortly introduce this text that is derived from the trilogue. But before that, let me shortly summarize some of our concerns in regard to this proposal. So based on the proposed criteria of Annex 1, companies could purposefully design NGT plants that can enter the market by fast-track approval, no matter about the degree of the environmental risks. This was explained on the example of a maize being with insecticidal properties developed with the help of artificial intelligence. And of course, you could have lots of other examples going to the same direction. And we already know that many companies are using artificial intelligence programs specifically to develop these plants. So this is really something like an invitation to those companies to purposefully design those plants to circumvent any risk assessment. If this regulatory proposal would be adopted, it would put a huge burden to the coming generations, but would leave them without adequate instruments to intervene if damage occurs, because if there is no risk assessment, there is also likely no detection method to be available. It will not be requested. So you cannot trace and track and monitor these plants and recall them from the environment if needed. And if the risks and the risk posed by the proposal, it's not only the environment as nature, which could be understood as nature, it's also agriculture, which is under risk. If non-tested NGT plants, such as release for experimental field trials, for examples, are introduced into the gene pool of breeders without notice. So what can we do to prevent these concerns to become reality? Next slide. This is now the text of the trilogue agreed in current negotiations. And in red, you would see the changes that we would suggest to this text. And this is now the first paragraph. It has 3 paragraphs. This Annex 1 criteria. And the first paragraph, we would say, let's forget about all these thresholds and just include a verification process, establish a verification process that is based on a molecular characterization that includes intended and unintended genetic changes. And let us compare this to the gene pool, which is already present or available for conventional breeding, and therefore characterize the plants and their similarity or the equivalence to conventional breeding. This is the only solid scientific method you can apply if you want to have established such a verification process to come up with some plants that then might enjoy fast-track approval process. So this would allow to identify those plants which you really have a higher likelihood not to go along with characteristics that are novel to the environment. Next slide. In the second paragraph, we see some type of modifications listed, and we basically would agree with it. But we also are aware that further definitions might be needed. What exactly, for example, is conventional breeding purposes to make sure that the criteria do not become expanded beyond what is actually present in the existing gene pool. And the third and last paragraph would, next slide, would deserve some more changes. So we think it's a very good idea to mention proteins, changes in proteins, intended and unintended proteins, but these chimeric proteins or novel proteins can not only be caused by the interruption of endogenous genes, but there are many other reasons and causes one could discuss that such proteins occur in NGT plants. So all these plants where these proteins are found should undergo risk assessment, and not only those where the chimeric protein is being caused by interruption of endogenous genes. Does not make sense to restrict it. Furthermore, we already heard that intervening with a gene regulation, with gene expression, is likely to be the most effective way to produce NGT plants with novel characteristics. So of course, also the gene expression and elements which are related to it should undergo risk assessment if the outcome is different to those if compared to conventional breeding. And finally, we also think it's a good idea to introduce the recital 13A as proposed by the European Parliament, which says that plants that can propagate and spread in the environment need specific careful measures, and therefore we would exclude those plants in general from becoming NGT1 plants. So that's our proposal. You can get it in writing, of course, when we send you the presentations. There is also down there a link to a text which is already online, which explains our proposal in more detail. And all we can say that we think that we all participants in here, and maybe all participants in and all members of European Parliament and the member states, all would agree that innovation can only be sustainable if the products, innovation only can be sustainable if the protection of health and environment is ensured, and therefore, please do not adopt this compromise text because it will not safeguard our future livelihood. Thank you very much.